Okay, then we'll just get into today's material. So last time you'll recall, we were talking a little bit about we've been talking about behavior and genetics, in particular, trying to understand the heritability of behavior. I provided you with a couple of examples of what we call artificial selection on behaviors, especially related to this nest building behavior that we talked about in class with mice, and how by breeding or arranging the mating of mice that have high and low nest building behavior, you can create a mouse. You can have a mouse evolve to produce a behavior that leads to a very high or a very large amount of nesting material. That is, that they create a large nest. So this kind of experiment shows that this behavior is heritable. That is, that parents can bequeath their behavior to their offspring through their genes. That is, that there is a genetic basis for some of these behaviors. So I want to follow along that line by asking questions about complex behaviors, in particular with humans. Of course, that's sort of one of the more compelling discussions about the heritability of behavior. And one area of profound interest is the idea of intelligence, and the idea that what is the heritability of intelligence? How do we define intelligence? I mean, these are all questions that are beyond the course per se, but I do want to just sort of introduce you to some of these ideas. And in particular, I want to discuss some of the controversies around these kinds of questions, certainly the way they are answered. And this is, this graph is a typical example of sort of the misuse of this kind of approach to understanding intelligence. In this case, you have an IQ test, which seems to indicate here that Black students have a lower IQ than white students. And of course, that's not the case. But the point here is that you have to understand what do we mean by intelligence? What was the test that was used to actually calculate IQ? Sort of this x-axis here is IQ score. What is that? How is that calculated? What was the tests that are used, and so on? These are all important questions that have to be addressed when we ask questions about intelligence. It's not an easy thing to answer, but the big question is about how do we get around asking questions about heritability and complex behavior? And one way to look at that is, obviously with people, we can't use selection experiments like what we saw with the mice. That would be ethically wrong. So we look for surrogate measures of heritability. So that is that we can do things like look at twin studies. So in this case, we can compare, monozygotic versus dizygotic twins and look at, what is the heritability of behavior when we look at these at twins. So the idea here is like monozygotic twins are basically identical twins. So that is that they are identical twins that share the same genome, and they are raised in the same environment. Whereas dizygotic twins are, twins that are born at the same time. That is that they don't share the same genes. That is that they're both from the same parents, but they're not identical. And so by comparing monozygotic to dizygotic twins, you can try to test hypotheses about, the heritability of complex behaviors. So, I'm not speaking specifically about intelligence here, but if complex behaviors are highly heritable, that is that if there's a high heritability between parent and offspring, then we would expect that monozygotic twins should have a higher similarity. They should be more similar to each other in terms of complex behavior than dizygotic twins, right? That's simply because monozygotic twins share the exact same genome. And therefore, if complex behavior is heritable, then we would expect those monozygotic twins to be more similar. If heritability is low for complex behaviors, then we expect that monozygotic and dizygotic twins should have very similar heritabilities. So again, we're not specifically speaking about, about intelligence per se, but these are just behaviors in general. And so this is the general pattern we see. So many of you will, this is the mainstream media often have stories about twins, right? There's always a story about two twins that were separated at birth. They were identical, and then they find them once they're in adulthood. And there's always some story about how, they have wives that are the same name, if they're men, of course. They may have partners that are the same name. They may have they have similar habits, even though they were raised in completely different environments. And this is sort of reflected in this here, right? These are twin studies. This is a broad result from twin studies. And these are twins that live together. But the point being here that monozygotic twins tend to be more similar than dizygotic twins. That slope that you see there in those graphs, that represents the heritability, or I should say the relationship between monozygotic twins and dizygotic twins there. And the steeper the line, the more similar they are. And so what this shows you is that in general, and this is not based on, this is a broad perspective that's sort of trying to give you a sense of how this works. But this isn't based on, it's based on data, but I'm just trying to give you the broad strokes of it all. The point here is that monozygotic twins tend to be much more similar than dizygotic twins across a variety of different behaviors. And so the point here is that there does seem to be high heritability for, and I say relatively speaking, high heritability for complex behaviors, specifically here in people and humans, but this is reflective of most other species as well. Now, this is a historical result from a guy named Hermann Siemens, who wanted to try to assess the heritability of intelligence in people. And so again, he looked at twins, and in this case, he was looking at their grades. So this was a proxy measure of intelligence, right? So again, we can be highly critical of this approach. This is from 1924. The point here is, this guy, Hermann Siemens, was his objectives were not necessarily, how do I put this? They weren't necessarily, just interested in the science per se, but there may have been motivations behind trying to determine why, or how intelligence was heritable. And we'll talk a bit more about that later on. But here, the point being that monozygotic twins tended to have more similar grades than dizygotic twins. So again, this is just not great support, but it just gives you a sense that the conclusion based on this was that there was a genetic basis to intelligence. That is, that there is, in this case, that there is some aspect of heritability. Now, having said that, and we're going to get into this a bit more later on, but this does not mean that, that genes control intelligence. There are genes that are associated with intelligence, and and whether this is, you use this data here, or there are others that we'll talk about. But I just want to be clear that that there is no sort of nature versus nurture aspect of this. The genetic underpinnings of intelligence are filtered through the environment, both the internal and external environment. And so I'll show you some figures in a second that will try to tease that apart. But the point is that, as you learned earlier, the phenotype is the result of an interaction between the genotype and the environment. And, in this case, intelligence is no different. So, so there's lots of caveats for these types of twin studies, to be fair. There's assumptions that, like I mentioned, the monozygotic to dizygotic twin comparison, the assumption is that the twins share the same environment. But that may or may not be the case, right? Depending on how you define the environment, each individual has its own sort of experience with the environment, and therefore you can't necessarily make that assumption, right? That in the case of humans, that these kinds of twin studies, that the twins share the same environment, at least in the kinds of examples I've just shown you. There are adoption studies that have also been done. And this is where twins, for example, are adopted out, and they are genetically identical, but they are raised in completely different environments. And so we can compare the similarity between offspring and their parents, the kids and their parents, and you can compare between genetic parents and with adoptive parents. And so if behavior, intelligence included, is highly heritable, then if you have a pair of identical twins that have been adopted out, then you would expect that they would be more similar to their genetic parents than to their adoptive parents. So that would be the case if the behavior or these kinds of complex behaviors are highly heritable. Now, if the heritability is low, that is, if there is very little genetic influence on these complex behaviors, including intelligence, then we would expect that that the similarity of offspring should be they should be more similar to their adoptive parents than to their genetic parents, right? Because that implies that the environment is more important. So here is, these are some data that look at correlations among different groups of parents and offspring and between sets of twins. And we'll just walk you through what they are. So the first here, so along the y-axis is just a correlation coefficient, right? So the higher the number, more correlated these are, the more similar they are. So this is parent offspring. PO is parent offspring when they are together. So this is your correlation. That's how similar they are. This is parent offspring when the offspring are adopted apart. And these are adoptive parents, the parent offspring when the parents are adopted. That is, I should say, they are not genetically related. These are parents who have adopted kids. And this is the relationship, the correlation for IQ, all right? Or I should say verbal ability. This is one aspect of IQ. So this is a standardized test that you can use, and that is the test of IQ, I guess, here is verbal ability. Then we have monozygotic twins that have been adopted apart, monozygotic twins that are together, and dizygotic twins that are raised together. Now, if we want to look at the relative effects of genes and the environment, we can look at these different groups and try to sort out, what are the different effects. So in this case, if we want to look at the effect of the environment, so here is the correlation between monozygotic twins that are raised apart and monozygotic twins that are raised together. And so these are genetically identical twins that have different environments, right? And that's adopted apart. And these are the monozygotic twins that are raised together. And this difference here shows you the effect of the environment. That is the difference between these two groups. They're genetically the same. They are monozygotic twins, but they are raised in different environments. Okay, and so that difference here between 0.51 and 0.76 gives you, that's the effect of the environment on verbal ability. Oh, I missed one. Oh, sorry, here. Crap. There we go. Okay, so, and then we can look at the effects of genes, right? The effects of genetic variation. And so on the left here, we have parents and their offspring. You can see these are parents raising their kids, their own kids. These are adoptive parents raising offspring that are not their own. And that difference of 0.28 to 0.08 reflects the effects of genes. That is, that there is a strong, there's a genetic effect. And the same here. So we have monozygotic twins and dizygotic twins raised together. These twins are raised together, and you have two categories, as I said, monozygotic and dizygotic twins. And this difference here is the result of genes. That is, that Dizygotic twins do not share the same genetic background, but these all share the same environment, and therefore this indicates here this difference is the effect of genes, of the genetic background. Okay, so the point here I'm trying to make is that it takes both the environment and the sort of genotype to affect the phenotype. In this case, the phenotype is a measure of verbal ability. And so intelligence, the suggestion is that intelligence has, we're not using IQ tests here necessarily. We're thinking about aspects of intelligence, whether it's verbal ability or other aspects, tends to have a heritability of around between 0.4 and 0.8, depending on the study and depending on the exact aspect of intelligence that you look at. And that is very similar to what we see in other complex behaviors in other species, right? And in other types of, other species in terms of verbal ability. I'm not talking about that. I'm talking about complex behaviors. But even like that kind of heritability is similar to what we see in heritability of like height, for example, and other morphological and physiological traits. So behavior really isn't all that different in terms of having a relative a relative contribution of genetics to explain variation in the trait. Okay. There are other effects that can, or other aspects that can affect intelligence and other complex behaviors. The maternal environment can be very important. So, exposure to hormones in the uterus, for example, can have consequences for a number of different behaviors, perhaps even intelligence. There is something called epigenetics, which is basically, these are genetic effects of the environment, and these can also affect behavior. And as I said, most behaviors that have been studied, including the kinds of complex behaviors like verbal ability, like I just described, have a heritability of around 0.5, which is the same as for, as I mentioned, other types of traits. Now, what I've told you is really not all that, shouldn't be all that remarkable. Yes, intelligence has a genetic component to it. That is that some of the differences in intelligence in a population, part of it can be attributed to, our genes, but also a large proportion of it can be attributed to our environment. Okay, so our internal environment or the external environment. And so the problem is that this kind of information has been misused, right? And we can reflect on that first graph. How do you even define intelligence? These kinds of questions. And what I just want to introduce you to here is some of the controversy around our understanding of the heritability of these kinds of traits, in particular related to intelligence. And this is mostly related to eugenics. And I brought this up in my evolutionary biology class, and some of you may have maybe in that class and have heard this already, but I'm just going to reflect a little bit again here for this course. And that is that a misunderstanding of what heritability means has in the past led to, drastic things like eugenics. So what is eugenics? Eugenics is the sort of like it's a policy in which evolutionary principles like our understanding of heritability are used to try to, quote unquote, improve the human race, right? So of course, the first thing that comes to mind is Nazi Germany, right? Nazi Germany, that there was the idea that, some groups of people, ethnic groups or otherwise, were carried with them traits that were undesirable. These were heritable traits, and therefore, there's the Holocaust and all of the horrors of Nazi Germany. Now, what's interesting is that those ideas of trying to improve human and I'll say the human race because that's the language they used back then, to improve the human race. To, you could do this by having or trying to support people to have kids. That is, the right kind of people should be having lots of kids. Or you can suppress, the wrong types of people, the people who shouldn't be having kids. And when I say these words, what I mean is that at the time there was sort of this idea that some people should have more kids because they are, in the case of Nazi Germany, you could talk about them being of the Aryan race, quote unquote, blonde, blue-eyed, intelligent and all this, and they thought others were less so. And therefore, people who were considered Aryan were then, told to have more kids and so on. But there's other ways to do it, and other ways that have been implemented, not just in Nazi Germany and the horrors that we saw there, but also even before Nazi Germany, across the UK, the United States, even Canada. There were all kinds of attempts to try to reduce the reproduction of people that were considered, quote unquote, undesirable. So even in the United States, there were academic societies that were devoted to the study of eugenics. The UK also, as I've mentioned, maybe not here, but certainly in my evolutionary biology class, there are certainly people, scientists that have contributed to our understanding of evolution and behavior who espouse some of these horrible ideals around eugenics. Even in Canada, we've had sterilization laws. So in Alberta in particular, people who were considered mentally defective were sterilized. And the idea here was that many, whatever mentally defective means, that because these, intelligence and other aspects of sort of human behavior were considered to be heritable, that is, that they thought there were genes that were dictating how people behaved or how smart they would be, that by preventing them from reproducing, that would improve the human race, whether it be the intelligence of it or what have you. So all kinds of academic, quote unquote, research about this. Here's, this is the Supreme Court of the United States upholding sterilization laws. 3 generations of imbeciles is enough. And in fact, there were terms used that were categories of how, and people were assigned to categories like imbecile and idiot. Like these were actual like clinical terms that were used. And the idea here was to uphold laws of mandatory sterilization of, quote unquote, mental defectives. Even if you, and I brought this up in my other class too, so apologies if you've heard this before, but even, the famous 5, and these are women who fought for the right to vote for women in Canada. Many of you will have seen the short video vignette that shows up on TV every once in a while. And then here they are glorified on our $50 bill. But in fact, many of these women espoused these sterilization laws in Alberta and other places. And so they were supportive of these kinds of eugenics, policies. And you do yourself a favor and read about what some of these women did in terms of what they wrote and what they were, and their opinions about some of the, highly racist, often very, supportive of eugenics. And, you should take a look and see what kind of people these were. Emily Murphy is one of the ones that is particularly, I'll say guilty of the time. The part of the issue is that at the time, some of these ideas were considered socially acceptable, right? So it's a very different time, of course, than now. All right, so this is, you can Google this and Google it. You can look up this stuff, but in Alberta, they were doing sterilizations of people who were considered mentally defective, and what that means, and who was, how do you define someone in that case? It's open to all kinds of interpretation, but certainly even in 1972, which is just, that's even after I was born, there were 2,800 sterilizations performed in Alberta under these kinds of eugenics policies. So, finally in 1972, later in 1972, this act, Sexual Sterilization Act, was repealed. But all to say that it's not that long ago that governments were pursuing these kinds of things, and all of this based on the misplaced understanding of what it means to have genes associated with particular behaviors. In this case, of course, we're speaking mostly about aspects of intelligence, and that's what was used as a basis for determining whether somebody should be sterilized or not. And even now, even now, this is 2004, which of course now is a bit getting old. But we still have arguments about academics trying to write and make arguments about the need or the value of doing this kind of, they're not calling it eugenics now, of course, but trying to make policy around assumptions about the heritability, about what heritability means for different behaviors. In particular, we can talk about, antisocial behavior, right? Violent behavior. So rather than intelligence, now people are arguing about these kinds of issues with respect to behaviors, like I said, like antisocial and violent behavior. So, the idea that genes can somehow dictate who is antisocial and who is not, and then, do you somehow act on that? There's some evidence, not evidence, but some people are arguing that you could intervene in terms of if you have people who are identified as having genes that are associated with anti-social or violent behavior. How can you intervene during their childhood to try to alter the environment to minimize the chances of that kind of behavior emerging? I mean, these are all very open questions. But I think the point here is that much of these the controversy around this is driven by the misunderstanding that the general public has about what does it mean to have a trait that has a genetic, basis. That is, it's heritable. And there's a distinct difference between the argument that the genes can influence behavior versus something that's called genetic determinism. And that's the genetic determinism is the point that seems to be used by people who espouse these kinds of eugenics type policies. Genetic determinism means that because you have a gene for behavior X, that means that you are going to manifest that behavior. And that's not true. Genetic determinism doesn't work like that. That is that if you have genes that are associated with a particular behavior, that association is filtered through the environment. And so, here's an example. So this is, this is E.O. Wilson. So E.O. Wilson is a very famous evolutionary biologist, studied behavior. He published a book called Sociobiology back in the in the 70s, And in that book, so the book was called Sociobiology, and it was basically doing what we have been doing in this course so far. Basically, he was doing like a tour of the animal kingdom and explaining how genes affect the behavior of animals. And, anything you were to read in those first chapters would be completely normal in the sense that they would be completely consistent with what we talk about in this course, what we've talked about so far, and what we will talk about in the future. But Wilson wrote, the poor guy, he wrote a chapter on humans. So what he was trying to say was that there are obviously some genetic influences on human behavior. And this was misinterpreted to mean that he was saying that our behaviors are genetically determined. That is, that there was a big debate because many argued that Wilson was saying there was no such thing as free will, for example, that we are completely driven by our genes, that our behaviors are driven by our genes. And this is this quote here, right, from one of Wilson's books. So E.O. Wilson expanded his chapter in sociobiology that was on humans, and he wrote this book called On Human Nature. And here, according to Wilson, human males have a genetic tendency toward polygamy. If true, then ladies, don't blame your mates for sleeping around. It's not their fault. They are genetically programmed. right? And so the point here is that this kind of misinterpretation of heritability of behavior leads to these kinds of faulty, perspectives. So it's not that, just because we in the historical or evolutionary past, maybe there was an adaptive value to polygamy in males, but that doesn't mean that we're living in a society where we can, that is, or should I say that the environment in which we are living necessarily means that all males are going to end up being polygamous. So, anyway, I should say E.O. Wilson just died recently. He was very old, but also incredibly prolific, and it's worthwhile reading some of his work. He was an expert on ants, actually, and sociobiology of ants, and understanding the eusociality that ants have. Anyways, he was. I have to say, maybe I'll find here. Hang on, just a second. So here, this is E.O. Wilson here. I don't know if you can see that. That's me, and that's E.O. Wilson. And in 1997, E.O. Wilson got an honorary degree from the University of Guelph, and that's where I did my master's. And he, anyways, I had the opportunity to meet him afterwards. And of course, like, what do I do, right? Like, I put like the rabbit ears behind him, completely disrespectful, terrible. I'm an awful person, but anyway, he's a very kind, was a very kind man, and very interesting, and lots of great work. But he's most well known for this controversy around his books. And it's worth reading about, in part about what happened, because one of the things that happened to him was when he was giving a talk about this work, someone poured like a container of water over his head. I mean, there was so much vitriol against him by his colleagues, even at Harvard, where he was, that and in the end he was vindicated, because the whole field of behavioral ecology is based on his insights, or at least partly on his insights. And anyway, at the time in the 70s, it was quite something. It was a very big debate. Okay, so all to say that there is, you hear the statement, there is a gene for behavior X, right? Let's say intelligence or whatever, verbal ability, emotional something or whatever. There is a gene for behavior X does not mean that the gene is fully responsible for the behavior. Okay, what that means is that there is some of the variation in the trait, some of the variation at the population level in a trait has a genetic basis. That is that there are, some individuals that carry, genes or numbers of genes that are associated with a particular behavior. Yes, that exists, but it's important to know that the environment plays an important role in all this. So in determining behavioral traits, it's not just genes, but it's also the environment in which an individual finds itself, and where the individual has developed, that are also critical to the ultimate outcome of behavior. Okay, so I guess, many of you will have heard of this nature versus nurture, quote unquote, debate, right? The idea that, that behavior, for example, is either the result of our genes or the result of our environment, one or the other. But that's not the case. It is both, right? So behavior is the product of continuous interaction between the genotype and a constantly changing environment. So the expression of behavior, the genetic expression of behavior, is dependent on the environment. So genes, as I've written here, genes provide a range of behavioral possibilities modified by the environment. So genes can be either, they can provide a behavior that's very broad and very flexible, or very narrow and fixed. Behavior is not programmed by the genes. And I guess that's the point I'm trying to make. Like your behavior is not programmed strictly by your genes. Your behavior is reflecting not just your genetic background, but also your environment, the environment that you developed in, your internal and your external environment. So the behavioral phenotype then is always susceptible to the environment. Yeah, so again, so just to reiterate some of this, so genes don't directly change the behavior. Genes produce proteins or other products like RNA. Any changes in a gene are going to change that protein, which can change cellular interactions, which ultimately, after a cascade of different things, leads to behavior. So it's just, my point is that it's just a very complex interaction between any particular gene and the expression of behavior, whether it's multiple genes, right, that produce a series of different interactions, sort of at the biochemical level, and then interacting with the environment. It becomes, you can't say that one gene, dictates a particular behavior. Having said that, there are lots of complex behaviors that are influenced by single genes. And so, those single genes interact with aspects of the environment, and they can lead to very complex behaviors. And what you're seeing here are just Drosophila sects. These are fruit flies, Drosophila melanogaster. We'll call them Drosophila for short. And there's this group of flies is a very well-studied group because their genes are fairly simple, and they've been heavily studied. And so scientists understand the genes of these flies, and they're able to assess their behavior in a very straightforward way, because their behavior is relatively simple compared to, say, a vertebrate. Can you guys hear this kind of whining behind me? Yeah, sorry, that's in my office. I can't control that. So anyway, you'll have to forgive me, but it's upstairs. Okay, so the one thing that fruit flies are really great because you can induce mutations in them. And when you induce mutations, what that means is that you change the genes. The DNA sequences are changed or altered. And by producing mutations, you can determine which genes are associated with particular, phenotypes. So for example, these are different. So first of all, they name these mutations, right? So they name the genes based on the behaviors or the phenotypic traits that they are associated with. So in this case, so there's one called LOT. So this is a gene that at this particular mutation leads to an elevated preference for salt. So the story of Lot, and you may be familiar with the Christian Bible. There's a story of Lot who turns to salt, or is turned to salt. Cheap date, which is somewhat misogynistic, I think, but increased susceptibility to alcohol. So this is a gene that's actually been given the name cheap date, and this particular mutation leads to the flies having more sensitivity to ethanol. Etheragogo, these are silly names. This is a gene that if there's a mutation there, the legs will shake when they're anesthetized. And there's a few others associated with reproduction, right? Stuck, mating with a mutation at this particular gene or locus means that mating males can get stuck while they're mating. Coitus interruptus, this gene is a mutation of it is related to brief copulation. And icebox, females not responding to courting males. Again, this is a gene that's regulating female response to courting males. And if there's a mutation at this particular gene, then females do not respond in this sort of normal way. Now, in terms of behavior, there is a very interesting behavior that's driven, and we'll show you in a minute, driven by a single gene. That is that, the single gene is important in the expression of this behavior. And this again is in fruit flies, Drosophila melanogaster. And it is relevant only to their larval stage. So this is, what is the larval stage of a fruit fly? You may have seen them if you have them in your house, but these are small sort of maggots that are running around searching for food. And they have two different behavioral phenotypes that occur in the presence of food. So this only occurs in the presence of food. And this is a behavior of the first called sitter. And what this is this is the path that the maggot, this larvae, may follow on an agar plate. So this is a circular plate that has agar, which is, I mean, I assume most of you know what that is, but it's like a gelatin that they sit on, and it's the same with the rover there. So, and what you see there is that the two phenotypes, the sitter, the larval fly, does not move very much, in contrast with the rover. And the cutoff here is the amount of movement. The way they classify this is based on the distance moved over 5 minutes. So in this case, if a fruit fly larva is moving less than 7.76 centimeters in 5 minutes, it's classified as a sitter. And if it's moving more than that 7.6 centimeters, it's classified as a rover. So here is just some data that reflects the differences between sitters and rovers. And you can see here, these are sitters, these flies. You can see the track length over 5 minutes. And you can see that you've got mostly, it's mostly dichotomous. That is that, there are some individuals that seem to sort of jump in between, but certainly you've got this sort of bimodal distribution that indicates that most flies are either a sitter or a rover. And the rover, if you take a rover female and a male sitter and you were to cross them, most of them are going to end up being rovers. And this is just trying to figure out what is the what is the pattern of inheritance of this trait. So first of all, it implies that the rover behavior, the gene associated with the rover behavior, is dominant. It's dominant. And the sitter allele appears to be recessive. And so if you take the F1 generation from these rovers, the F2 generation is 3-to-1 rovers to sitter. And this is consistent with these Mendelian ratios if the rover were to be dominant and the sitter were to be recessive. So it appears that they've got a single gene where you have two alleles, a rover allele and a sitter allele, and the rover allele is dominant. And so this gene seems to be affect behavior in particular only with the larvae and in the presence of food again. Okay, so the environment, the developmental stage and the environment are influencing the behavior that is being expressed here. Now you could isolate the different alleles. So they have two, they call this gene FOR or 4 for the foraging gene. So that's the name of the gene that they've given it. It has a recessive and a dominant side. So it's got a rover and the sitter allele. The rover allele appears to be dominant. And you can actually take, I haven't shown you all this, but they have been able to identify this foraging gene. They've been able to insert that gene into these fruit flies, and they can turn a sitter into a rover by inserting this gene. So this gene then does have, they clearly has an effect on behavior. So again, based on this sort of pattern, they've, the rover, is that behavior is expressed by a homozygote and a heterozygote with this foraging rover gene, and the sitters, only the sitters that are homozygote express that sitting behavior. Now, they've been able to identify the gene product of that gene. So this foraging gene codes for something called Cgmp dependent protein kinase. We'll call it PKG for short. And this is an enzyme that's expressed in the brain. So these flies that have the rover allele have high levels of this PKG. And the sitter flies have relatively low levels of this PKG. And you can actually take a larva that is a sitter, that is, it's genetically, it has the sitter allele, and you can dose their brain with this PKG, and they will shift to a different behavior. They will shift to the rover phenotype. So this single gene then does influence the behavior in the presence of food, as I mentioned. So the environment, that's the influence of the environment. So the question then from our perspective, we're interested in the adaptive value of behavior. And so, we ask the question, what are the fitness consequences of this gene and the behavior that it, seems to influence? And so you can take a population of 50% rovers and 50% sitters, and you can have two, in this case, they had two treatments, a low fly density and a high fly density. So that means that the density of the larvae was either low or high. And you can test to determine, which is going to emerge as the most predominant phenotype, the sitter phenotype or the rover phenotype. And so, you might ask, well, why does density matter? Why are we manipulating density? And so if food patches, under certain circumstances, the sitter larvae may remain on a food patch longer. And if they do that, then that might be more costly at high densities of larvae or of flies. And in contrast, the rover larvae might be able to find a new patch much sooner than a sitter larvae might. So at high densities, it might pay to be a rover to basically run around and be able to find more food. And so what they did was that they took these two different treatments. Now, fruit flies are raised, at least in the lab, they're raised in vials. The vials are provided with food, and you can manipulate how many flies are in these vials. And in this case, there were two treatments, 50 flies per vial in one treatment and 800 to 1,200 flies in the second treatment. So clearly low density and high density. They allow these flies to breed for 74 generations. And we would expect, based on the argument that I just made in the previous slide, we would expect that the high density flies, even though they both start with 50% rovers, 50% sitters, we would expect that the rovers would have an advantage in the high density scenario. And therefore we should expect to see a predominance of the rover phenotype in this, after these 74 generations in the high fly density. And that's what they find. So there's three sets of lines that they look at, that they use to replicate them. Here along, so that's what's along the x-axis here. The path length here gives you a sense of, rover versus sitter. The rovers are going to have a higher mean path length than the sitters. And you can see that under the low density, we see that the sitter type is phenotype is favored. We see that the mean path length is much shorter, consistent with that sitter phenotype. And in the high density phenotype, sorry, high density scenario, we see that the mean path length is much higher, indicating that the rover type is favored under those situations. So there is an adaptive value to these different behaviors depending on the density of the population, right? So high density seems to favor the rover type, low density seems to favor the sitter type. So again, these are the explanations, right? Perhaps the rover type is favored in the high density situations because they can find food more readily. And if the densities are high, that means that food might be at a premium. That is that there's lots of other flies or larvae that are running around, crawling around, looking for food. And so it's adaptive then to be looking for more food constantly through, by traveling further. Versus, the low density lines, which seem to favor the sitter type, the sitter phenotype. And perhaps because you have to waste less energy to search for food. So there's no point running around looking for food if there is more food available per capita. So per larvae, there's more food available because it's low density. And therefore, you'd be doing nothing more than wasting energy if you were running around looking for more food, because you could just sit in the same spot and feed. So the rover, this experiment shows that behavior can evolve. That is, that you can see a change in the fitness of the different behavioral types in this case, right? The rovers and the sitters. And you get a change in the preferred phenotype, right? So that's evidence of evolutionary change. The behavior is, the average behavior is changing as you move from generation to generation. And after 74 generations, there is evidence that different, in this case, different densities favor different phenotypes. Now, there is a gene for behavior X does not mean that the gene is fully responsible for behavior X. So in this case, the rover and the sitter larvae behave differently only when food is present. So they have very similar behaviors when when food is absent. Okay, so when food is absent, you'll see that the pathways are very similar. In this case, of course, they're longer. Whereas when there is food present, that's when you see evidence of behavioral differences. So the environment is very important in terms of seeing the manifestation of that behavior. So rover larvae will behave like a sitter if starved prior to exposure to food. And there also is evidence that there is a modifier gene that can affect the expression of the rover behavior. So there's a whole series of different, both the external environment, but also aspects of the internal environment or internal conditions that can affect the expression of the behavior. I'm going to stop there, but what I'm going to do next time is talk a little bit about how we can look at similar genes in other animals and see how that this gene modifies the behavior of those different animals in very similar ways. So we'll talk about bees and the differences across different types within the bee colony and so on. All right, so we'll stop there. If you have any questions, let me know. Otherwise, we'll see you on Thursday.
Thank you.
Yeah.
Thank you.
Sure.
Thank you, Professor.
Thanks, sir.
Thank you, sir.